If you have any questions and answers, or if you have any questions, or if you have answers, there's mics. There's one mic over here. That's the one. So please come up and ask them. And I just want to draw your attention, which I think is really a nice part of the program, is there's a whole list of resources that have been developed to explain stem cell research and some of the issues. So it's on the inside of your program. And also you received an evaluation, and these are really important in terms of planning additional programs and also for grant funding. So if at some point during the day, if you wouldn't mind filling it out, and there's a box in the back. So I will let you handle the questions. Okay, we'll just...
Hi, my question was around the two 10-year goals that you mentioned.
I'm sorry,
the two 10-year goals that you mentioned. So the first one, if you could clarify that, because I didn't really understand. I understand phase 1 and phase 2, but I don't understand what can restore function actually constitutes. What does that actually mean?
Right. So I think these goals are attempting to be as specific as they can be, given that there's a lot that we don't know. But the overall goal is that at least for one disease, we will get to a place where the use of cellular therapy can be applied in patients. And I think that's the bottom line of that goal. Does that help clarify it at all?
So beyond phase 1, phase 2, all the way to the point where it's actually being...
Yes.
Hi, I just had a quick question about the first couple of slides. You showed them, I think it was called the
Pluripotent cells?
pluripotent,
Yes.
and then it went down to multipotent.
That's correct.
Wonder what the difference is between those two.
Well, it's a definition or use of terms. So pluripotent means that a stem cell then is able to create all the different cells that are part of an organism. So if we have about 200 different cell types in our organism, that pluripotent cell should be able to create every one of those. A multipotent stem cell is one that has a more limited capacity. So it might be able to generate, for example, all the cells that are part of your nervous system and brain, but just those. And that's...
Makes sense.
Okay.
Hi, Gil. Mohamed Majdoubi, Dominican. Thank you for a wonderful presentation. My question is regarding the training. As you know, we're involved at Dominican in training undergraduate students in the goal of providing highly skilled technical assistance for the ventures and all the companies and institutions that are going to join California. My question is, the funding for training has been restricted so far to graduate level, pre-doctoral, postdoctoral, and clinical. And my question is, do you have any scoop for me in terms of undergraduate training? Do you know of any funding initiative coming in the short term?
Yes, the strategic plan describes an initiative for the development of technical training. And such a program, I imagine, would probably include undergraduate training. So that kind of initiative has not yet been fully developed, but it's something that we intend to work on. And it may come in parallel with the next 3 years of the training program for postdocs, pre-docs, and clinical fellows.
Awesome. Thank you.
You mentioned 2 to 4 therapy possibility diseases, and then one that you would be working on initially. And I noticed you had pancreatic cells in one of your illustrations. And so I hope that it will be diabetes. Thank you. Will it be, by any chance?
So what was the answer?
I don't know.
Oh.
But I think we can all hope for, I think what we want to do is have the science tell us what it is that will be ready, that will be mature enough to get to patients. And so that's something I cannot predict.
So I'm wondering how long in the adult do stem cells just continue, do they continue forever? Or is there a time, say, in aging when they actually stop being functional?
That's a great question. I think the answer to that is that in part we don't know, and I think it's a question that you may ask of Renee or Mohammed when they get up here as well. But there is that capacity that as we age and as we grow, stem cells do become more specialized. So we will always have, for example, the capacity to renew all of our blood cells, and it is the reason bone marrow transplants or bone marrow stem cell transplants work. Because of those stem cells that still exist in the adult. But we might not perhaps still have within us stem cells that can produce every cell of our body. So as we grow, they will very likely become more limited in their capacity, but yet enough to do what they have to do in order to make our organism work.
So also the new evidence of brain plasticity and ability to regenerate nerve cells, which we didn't think we had, that's related to stem cells?
That is related to stem cells, and I think that's part of the discovery through which, as scientists continue to explore what it is that stem cells do and what they're capable of, there are reports in different tissues that stem cells may exist. But I think the prime examples really continue to be those in the bone marrow that give rise to blood cells, and also in our gut. Our intestinal epithelia turns over quite a bit, and so you have stem cells that continually renew those epithelial cells. So there are very likely stem cells throughout our body that do that and maintain our body, but there may be other areas where that is more limited. And so there is probably a spectrum, but I cannot say that every tissue that we have will necessarily have a stem cell that continually renews it.
There have been other countries that have not been inhibited politically in moving forward with stem cell research, and they have not lost almost a decade of time. Are you integrated with them, and are you launching on their work to move ourselves forward faster in spite of tremendous inhibitions? And how are you doing that? With whom are you doing that? And how is it helping you?
That's a great question. So let me start off by saying that in general, scientists who work in any field don't tend to restrict themselves to those that are found within their institution, within their state, or within their country. They tend to collaborate and reach out internationally, and I think that's no different for stem cell biologists. So that's one aspect of it. The other is that the CIRM, very interestingly, was nominated to become part of the International Stem Cell Forum, which includes participation of many different countries who are engaged in stem cell research, who are trying to standardize efforts, take into account social, ethical, and legal implications of the work. And the United States was not, per se, made a part of that, but California was. So California has, in essence, become the representative for the United States, at least in that organization. And we have had several efforts to promote collaborations internationally. Many of our grant reviewers are not, well, actually, none of our grant reviewers are from California. They are outside of California in the United States, but also international. So we get their contribution as well. And we've also been a part of programs. One example was a California and United Kingdom event where we had and sponsored several California scientists to meet with several UK scientists to forge collaborations and to bring about sharing of their data and their findings to spur that on. But that is definitely a very critical element of what we do. I think even though Prop 71 is about California, I think the Institute realizes that this effort is an international, worldwide one, and it is not one that should be restricted to California.
Thank you. My question is kind of similar. I thought I heard you say that the stem cell research cannot be done in institutions that have NIH grants. And that would sound to me as if you can't work with UCSF scientists, Stanford University scientists. Are you able to give them grants, and how is that working?
Right. So it's that again is also kind of a murky area, and it's not that institutions that have NIH grants cannot conduct the research, but rather there is a question of whether institutions whose building facilities or equipment which were purchased with federal funds can be used to conduct human embryonic stem cell research. And that is human embryonic stem cell research that would involve the creation of new lines, those lines which were not already previously generated before that presidential executive order. So there is human embryonic stem cell research going on that in part is sponsored by the NIH, but it is only that which meets the executive order's requirements. And different institutions, I think, are interpreting this prohibition differently. There are institutions which have considered it an accounting issue, where they essentially separate the time that they utilize for a particular piece of equipment or lab space and say, well, this particular slot of time is utilized for the creation of new lines, and it's sponsored only through, for example, CIRM funds, and the other is through NIH. So I think, again, it's getting creative, and people are doing different things to get around it.
So you are able to fund.
And so we are able to fund research. Yes, the CIRM itself has no restrictions on the use of the money. Sure.
I want everyone to be able to get their questions answered, so we'll continue with the questions if you don't mind. But I also wanted to, we do want to start the panel right at 10:45. We need to kind of stay on schedule for lunch. So if you want to take a break, I just wanted to offer that to you. But but part of our goal is to get people's questions answered. So anyway, we will start the panel at 10:45. Okay.
Thanks.
Hi. If I'm an entrepreneur trying to advance the research side of the business, but I'm torn whether I should go for the funding request either as a researcher or as a for-profit organization, how do you judge that? Like, are my chances of getting the funding any better if I go the research route versus the for-profit? And also, what heuristics go through your mind or the international committee's mind when it comes to funding? Thank you.
I think when we put out an initiative, it has very specific objectives and goals. So, for example, one of our latest initiatives is on the development of new stem cell lines using new technologies or traditional technologies. And the research is what, and the research proposal is what the reviewers will look at most. Whether it comes from a for-profit company or whether it comes from a university, if it meets the objectives laid out in the initiative, that is what will be deemed the most scientifically meritorious. So it really is about the project, about the research, what the proposal is, and I think less so about whether it is a for-profit company or not.
Hi.
Hi.
I did my PhD with Roger Peterson, who you know has left the country to go to England and start stem cell research there. My question is, do you know of or have any knowledge, or have they come back to you or given any feedback of the candidates that are now running for president, whether they would be proactive in the stem cell research area, embryonic or adult?
They have not, at least certainly not with me. I think there is the general belief among many people that regardless of what happens in 2008 and 2009, that we will probably see an easing of the current federal restrictions. But even with that, I think it's also important to note that currently the National Institutes of Health has been, on a yearly basis, cutting its budget. And so the support for stem cell research, although it may open the door for certain research proposals, will not necessarily increase. So it is still important that the state efforts continue in order to see this research through.
Hi. Given the uncertainty of this new area in science and given the amount of money that's being put into it, how certain are you that our money will not be wasted?
Well, that's the role of our institute. And the very reason why we need to consider all of these other elements that contribute to the research, the ethics, the social implications of it. But it's, I think, like any research endeavor. It is research and it is a question. And so it really is, you know, do we want to explore it or not? And if we don't explore it, what do we miss out? And I guess until we get there, we don't know. But I think the potential for it as an enabling technology has excited scientists like no other in recent times. So I think it's one that does hold a lot of promise, but exactly what will be derived from it has not yet been completely clarified.
In England, there has been research done over the past, I'm going to say 15, 20 years, using our own white cells and retro-differentiating them, bypassing the need to use embryonic stem cells. I don't understand why that's not being looked at here. I also know that they are offering clinical work being done. You can go and have this done, and they're getting very positive results. And it's already happening, and people are already benefiting. I've talked to actual patients who have done this, and it's it's non-invasive. It's unfortunately a little costly at this point because it's so new. But you're our prime research group. Why are you only focusing on embryonic stem cells?
Okay. We are not only focusing on human embryonic stem cell research. I think it is human embryonic stem cell research which has been prohibited and therefore has, I think, suffered the most in terms of having support in terms of funding. But I think, you know, the question you ask is very key and important, because when we look at what the potential of stem cell research is and could be, we do have to look at all the different stem cell types. And there are many ongoing projects that we are currently supporting that utilize hematopoietic stem cells, that is blood stem cells, as well as human embryonic stem cells. And there are there's a very recent technology or discovery that I think has also impacted the science. That is, there are several groups that have shown that they can take an adult cell, like a skin cell, and been able to reprogram it so that it behaves very much like an embryonic stem cell. And so I think there is a lot still that we need to learn, but I think we can't learn it if we decide to focus only on one thing. So I think you're very right. We need to reach out not only to human embryonic stem cell, but adult cells and the new and different technologies that are coming into play. I think that's how we get to our goals.
Are you familiar with I'm sure you are, I assume you're familiar with taking white cells and retro-differentiating them?
There are different types of proposals that do different things that utilize bone marrows of different types. There are mesenchymal stem cells, for example, and then blood stem cells in which there are attempts to do those kinds of things.
Thank you.
Hi.
Hi.
Do you have clinical studies going on, and where will I volunteer? I am a Parkinson's patient. I'm also a cancer survivor, and I would like to volunteer.
So we are not at the moment at the stage where we are funding clinical studies, but we do hope to get there at one point. But when we fund these studies, these are organized and led usually by the institution or the individuals that we fund. So in terms of how you would participate and how you would get involved is really kind of keeping an ear and an eye open for possibilities that may be coming out of UCSF or UCSD or any of the institutions who may be conducting these clinical studies.
I think speaker deserves some coffee break now. Thank you.
Thank you very much, and you do deserve some coffee.